Good day, everyone. I'll present to you our study, K&Sick Pests and Weeds in Irrigated Rice Fields at Varying Fertilization Regimes. My co-authors are Rowena P. Varela, Elizabeth Parac, and Reuben James Rollon. This photo shows where our study was conducted. The background of the study, the world population is growing day by day and it is estimated to exceed 9 billion in 2050. Problems with crop pests challenge food security and poverty elevation in southern Philippines. Rice cultivation is commonly encountered by different biotic factors, including insect pests and diseases, which adversely affect its yield per unit area, according to Parac Rice Team 2017. Managing weeds in rice is tedious due to its expensive nature and labor scarcity. According to Surit Kumar 2016, at the local scale, the low rice yields in the Parac region have been closely linked to the rising population of insect pests and weeds in many farms across 5 provinces, according to Rice 2020. The objectives were described the occurrence of pests and weeds during fertilization at different phases of rice in irrigated rice fields, evaluated the soil characteristic of 4 irrigated sites, assessed the nitrogen and potassium concentration of rice crops in relation to pest occurrence, analyzed the correlation between the occurrence of pests and weeds against soil characteristics. The study was conducted in irrigated rice fields in RTRomualdez Agusan del Norte from July to December 2020 cropping season. 4 farmers' fields were used as sampling sites. 5 sample quadrats were selected randomly in each field. Collection of insect pests and weeds from the paddy fields every 15 days, and then between 6 to 8 in the morning after transplanting until the rice crop at the harvest stage. A 50 square meter frame served as the main tool to generate estimates of abundance, species richness, and percentage cover. A sweet net was used during sampling, which measured 8 inches in diameter, 12 inches in width, and 15 inches in height. And 180-degree arc counted 1 sweep up to 5 pass left and right in a 5 square meter quadrat. 5 samples were taken randomly per site in a zigzag direction. The collected insects were placed in transparent polyethylene plastics, tied with a rubber band, placed in the freezer for 3 hours to kill the insects, and removed from the freezer for identification. The collection of weeds was done randomly at each site. 5 samples were collected in a zigzag direction. Collected weed samples were placed in transparent cellophane and brought to the processing area for weed identification. Weeds were identified using the reference booklet titled Weeds in Irrigated and Rain-Fed Lowland Rice Fields in the Philippines by the NIRI et al. 2016. K&Sick pests were identified through visual examination using the reference titled Common Insect Pests of Rice under Natural Biological Control by Pepper 2015. Soil sample of 4 sampling sites were gathered randomly. Samples were taken per site and replicated 5 times. The soil samples were air-dried and submitted to regional soil laboratory to assess the soil pH, total organic matter, soil extractable phosphorus, and potassium. At harvesting, rice was cut randomly at the base and thrust, and rice rows were collected with 5 samples in each field to assess the nitrogen and potassium concentration. Quadrat sampling method was used to determine insect pests and weeds occurrence in the sampling area. A 50 square meter frame served as the main tool to generate estimates of abundance, species richness, and percentage cover. This was further employed every 15 days after transplanting. The rate of fertilizer application was taken through an interview with the farmers based on their actual application on their fields. The data on weeds and insect pests was analyzed in relation to the fertilization scheme. Data on diversity, abundance, and population fluctuation was analyzed using the multivariate statistical package. The figure presents the estimated occurrence or population of pests in irrigated rice fields at different fertilizer regimes. Brown plant hopper, green leaf hopper, and whorl maggot are common pests in the 4 sampling sites. During all stages of the rice crop, it is observed that the number of pests grows along with the application of different fertilizers such as ammonium sulfate, complex fertilizers, and urea. As shown in Figure 2, Site 2 was applied with higher amounts of nitrogen and other fertilizers and revealed a higher incidence of pests. This figure exposes the identified weeds in 4 irrigated rice fields. Common weeds species include Varying dichotoma, Rottboellia, and Echinoclea glabricens. Extreme cases of Varying dichotoma are observed in Site 1 and 3. According to Vawa et al. 2014, weeds shows a variety of responses to different fertilizers under different tillage systems depending upon the rate and method of application. Fertilizations alter soil fertility, thus affecting weed density, nutrient uptake, and biomass yield, which in turn affects species composition and biodiversity. Varying dichotoma grows well on wet or most likely in irrigated rice fields which maintain a 3 to 5 cm water depth, especially during the reproductive stages of the rice plant, in which this type of ecosystem is more favorable to Varying dichotoma to reproduce. Donaire et al. 2018 also reported that Echinoclea glabricens is highly competitive, especially when a seedling grows with rice seedling during transplanting or after direct seeding. This table presents the soil characteristic of 4 sampling sites in terms of pH, percent organic matter, phosphorus, and potassium. The pH levels in all sampling sites fall within the acceptable range from 6 to 7.5, according to Ilagan et al. 2014. The suitable soil pH for rice cultivation is pH 6. This implies that most nutrients are available for most plants in the mentioned rice fields. The percentage of organic matter in soils range from 2.82 to 3.28. This shows that soil organic matter across fields is in productivity levels. The largest percent organic matter is found in Site 2, while the least is revealed in Site 1. According to Well and Brady 2710, most productive agricultural soils have organic matter between 3 and 6%, wherein the soil organic matter of 4 sites is in acceptable range. Phosphorus and potassium contents and ppm are also presented in Table 1. Ilagan et al. 2014 also reported the range of 5 to 10 ppm indicates a medium level of available phosphorus, whereas high available phosphorus is greater than 10 ppm. The beneficial availability of phosphorus in soil is within the soil pH of 6.8, according to C et al. 2015. It is also noted that soil phosphorus across farms is within the desired value of 25 to 50 ppm. It can be gleaned that Table 1 that the concentration of phosphorus is higher in Site 1 with 47.40 ppm and Site 3 with 48.20 ppm compared to the other 2 irrigated rice fields. In terms of potassium content, Site 2 records the largest potassium content with 117.80 per pm. Available K ppm for rice ranges with values 20.8 ppm, in which the 4 irrigated rice fields possess a high level of soil K ranging from 98 ppm to 117.8 ppm. Although Site 2 has the largest potassium content, still added potassium fertilizer to his farm. ODHA RB et al. 2021 stated that the availability of potassium in the soil is proportional to the added potassium fertilizer, but a higher rate of potassium addition results in fixation rather than availability. Table 2 shows the concentration of nitrogen and potassium of rice crops in relation to pest occurrence. Data shows that nitrogen concentration is very low, which ranges from 1.01 to 1.10, and K concentration is marginally ranging from 1.03 to 1.26. It is shown that Site 3 postulates the largest mean nitrogen concentration of 1.1, along with the largest mean nitrogen concentration and third largest potassium concentration that is recorded in Site 3. It also observed that occurrences of common pests such as brown plant hopper with 23.6, green leaf hopper with 22, and whorl maggot with 10.4 are less dominant in the same farm. The other three sampling sites of rice field unveil a similar mean nitrogen concentration of 1.01. On the other note, Site 1 has the largest potassium concentration, as evidenced by a mean score of 1.26, also shows fewer dominant pests, which recorded 25.6 brown plant hopper, 19.6 green leaf hopper, and 14.8 whorl maggot. While Site 2 has the less mean potassium concentration of only 1.03, data showed that the farm received higher input of nitrogen and potassium fertilizers. On the contrary, a lesser concentration of nitrogen and potassium was observed in Site 2, where high dominant pest occurrences are found. Increasing levels of nitrogen and potassium application to rice plants resulted in a significant increase in nitrogen and potassium content in plant tissues, but on the contrary, it showed lower concentration of nitrogen and potassium. According to ODHA RB et al. 2021, the availability of nitrogen in the soil is proportional to the added K fertilizer, but a high rate of K addition results in a fixation rather than availability. In addition, K mobile elements and additional K results in leaching under flooded conditions in rice cultivation. This table shows the correlation analysis between the occurrence of pests and weeds against soil characteristics. It can be gleaned that of all the pests presented, the brown plant hopper showed a positive and strong correlation with organic matter and a strong negative relationship with phosphorus content, as evidenced by the respective person R values of 0.98 and negative 0.96. The incidence of DPH was directly proportional to applied doses of nitrogen and inversely proportional to phosphorus. In the study of Singh et al. 2018, DPH population showed a positive correlation with nitrogen levels in plants and a conversely negative correlation with phosphorus. Likewise, the occurrence of green leaf hopper is similarly explained by the organic matter of person R 0.97 and phosphorus content with person R is equal to negative 0.97. Rottboellia cochinchinensis is positively correlated with nitrogen concentration. Nitrogen remains the most limiting and important soil nutrient, according to Saito et al. 2019. Studies have also shown that increased nitrogen could enhance weed dry matter accumulation capable of competing with rice crops when weeding is delayed, according to Kodo et al. 2020 and 2021. In summary, the pH levels in all sampling sites fall within acceptable range from 6 to 7.5. The percentage of organic matter in soils range within 2.82 to 3.28, which vary across 4 sampling sites. Phosphorus is higher in Site 1 and 3 compared to other 2 irrigated rice fields. In terms of potassium content, Site 2 farm records the largest K content of 117.8 ppm and followed by Site 1 weeds 110.4 ppm. Brown plant hopper, green leaf hopper, and wall maggot are identified as common pests existing in the 4 sampling sites. Common weeds species including Fimbristellis dichotoma, Ludwigia octobulbis, and Echinocloa glabrescens are found in the sampling sites. Brown plant hopper, green leaf hopper, and wall maggot are less dominant in farms with higher concentration of nitrogen and potassium. Lesser concentration of nitrogen and potassium was observed in a specific farm where highly dominant pest occurrences are found. Occurrence of plant hopper showed a positive and strong correlation with organic matter and strong negative relationships with phosphorus. Occurrence of green leaf hopper is positively correlated with organic matter but negatively correlated with both phosphorus content. Rottboellia cochinchinensis is positively correlated with nitrogen concentration. Based on the findings presented above, the following conclusions were drawn. The soil characteristics of the sampling sites are on normal or considerable range. Occurrence of pests and weeds is evident in the sampling sites, which can be attributed to the presence of potassium and nitrogen. Plant hopper and green leaf hopper grow in areas with higher organic matter but less dominant in decreasing phosphorus content. Rottboellia cochinchinensis grows more when nitrogen concentration is increasing. That's all of my presentation. Thank you.